Imagine you receive a letter from your friend. He made a trip around the world. You don't know exactly where he is. Then you receive a letter. "Hi," he writes. "I'm here in a nice country. Many lovely people care for me, and I'm really glad to be here and to enjoy the nice mountains, lakes and seas. Hope to see you soon." After a time, you get the second letter with the same sequences and sentences, but different characters are marked in red. If you line up the red marked characters in this message, a complete other information receives you. Attention! This message was a fake. I'm in prison here. Please call police immediately. This exemplifies epigenetics. You have a superficial grammar of sentences which transport a meaning, but with the marking of certain characters you may change the meaning of the message completely. This is how epigenetics works. Every cell of every organism contains the whole genetic information on its DNA. Nevertheless, complex organisms such as humans have very different classes of cells and tissues, such e.g. muscles, synapses in the brain, liver cells, lung cells, skin cells and many other cells of specialized organs. They all function within their community system until they die, and others are reproduced at the correct place and time to replace the died cell, and the whole function of the organ remains healthy. Now it is possible to coordinate the reproduction with high fidelity of this constant exchange. How is this possible? It is possible through epigenetic markings that are inherent to all different cells. The epigenetic marking functions as a designation of the specific identity of each cell in every tissue organ of an organism. If a cell is damaged or is dying for various reasons, such as disease or even just because they are old, this circumstance is noticed by the cells around these specific cells by specific signals that are not present in a well-being cell. This triggers the surrounding cells to reproduce a new cell, and epigenetics ensures the exact time of this reproduction process and the exact place. Epigenetic markings can be inherited or not. It depends on how strong is the marking by the chemical components. So if an organism has a certain experience in a social way or in the body health way or environmental circumstances, this may influence the epigenetic status of the organism or several key tissues. This means an organism can mark an experience he made, and if this experience helps to learn how to better adapt to the changing environments or changing social interactions, and maybe it is also inherited. The next generation has a genetic program that better fits to environmental circumstances. And this is the way how evolution learned to learn, because species or members of species are not in prison to reproduce always the same pathways, but may find new ones, and these new ones can be inherited. The epigenetic research demonstrates that the central dogma of molecular biology, that the information flow in any case goes from DNA to RNA and protein, which was the dominant narrative in the 20th century, that this narrative is wrong. The correct way is also experiences of the body can influence how genes are regulated, constructed, and maybe also inherited. Where does this epigenetic markings evolutionary arose? Where does it come from? What is its history? The history of epigenetics starts with the virosphere, with the beginning of virus evolution nearly 4 billion years ago. All viruses mark their genomes to be as identity markers, to differentiate between self and non-self. With the rise of cellular organisms nearly 4 billion years ago, infectious agents constructed the genomes of cellular organisms by infection events and their counter-regulating parts. And therefore the epigenetic markings was transferred early at the beginning of cellular organisms to the cellular life on this planet. It is important that the identity of a cell is fixed by its epigenetic imprinting. So the whole genome contains the whole information, genetic information of an organism. But the various cell types are defined by their epigenetic markings. It is a liver cell, brain cell, skin cell, muscle cell. Less epigenetically fixed cells are the stem cells. Therefore, stem cells are the most appropriate tool to renew damaged tissue, because the epigenetic programming is not just fixed as in adult cells.